So, this is the last in our mass spectrometry training series, and today we'll be looking at applications of MALDI mass spectrometry. So what we'll do is we'll first of all look at what is MALDI. We did have a look at MALDI instruments right at the very start of the training program, but it's useful to have a look at the ionization method itself. And for MALDI, it's important to understand the matrices, what they're for, and which matrix is appropriate for what you're analyzing. I'm going to have a look at the differences between MALDI and electrospray, and give you an idea of what these differences are and how they the two techniques are suited to different purposes. And we'll look at the practical advantages and also some of the disadvantages of MALDI. I'm then going to focus on three different techniques. Firstly, MALDI applied to clinical diagnostics, which I think is extremely interesting. MALDI applied to imaging, and two different imaging technologies. One is mass cytometry, and the other is imaging mass cytometry. Okay, so MALDI stands for matrix assisted laser desorption ionization. The technique has been around for a long time, and essentially what happens is you will start with a liquid sample, usually very often a biological sample, but it doesn't have to be, and this is mixed with a matrix. We'll talk about what the matrix is in just a moment, but it's mixed with a vast molar excess of matrix, and it's then spotted onto a stainless steel sample target. I've shown you a just get my pointer. This is a sample target. This is a microtiter plate format, so it's a similar size. And each of these spots on the target are about 2 millimeters across. So there is a vast molar excess of matrix. So we have on the target we have a solid sample, and when the matrix and the sample have crystallized, we have a solid sample and matrix crystal, and these can then be introduced into the high vacuum of the mass spectrometer source chamber. So we have a UV laser, that's the laser desorption part, and the crystals are steered onto that laser beam, and the laser energy will cause ablation of those crystals. There will be desorption of the sample and of the protonated matrix ions into the vacuum. What then happens is the matrix ions are charged, and there is a gas phase charge transfer, which will generate singly charged analyte ions. In general, MALDI generates singly charged ions. That's not always the case. Occasionally, for a very large analyte, you may see two charges, but seldom any more than that. It's not at all like electrospray. And MALDI is an ion source, and like most ion sources, you can couple it onto any type of mass spectrometer. So this is essentially the MALDI process. Here's your sample introduction, which is very simple. We have solid sample introduced into the vacuum. It's irradiated with the laser. We have this desorption and then ionization happening. And I just thought you might like to see this. So this is MALDI acquisition in real time. So these are the spots on the target, and this is the laser being steered onto the spot. The laser is being fired, and the ablation will generate your spectrum. So let's talk about the matrix. What is the matrix? I borrowed this from one of my favorite films. It's quite old now. But this is Neo from The Matrix. He says, "I'm trying to free your mind, Neo, but I can only show you the door. You're the one that has to walk through it." Yes, very good film. But what is the matrix? So matrix molecules are small molecules that can undergo. So some molecules can undergo matrix-free analysis. So there is a technique called laser desorption ionization, and for small molecules you can dispense with the matrix altogether. But for large molecules, they will not ionize unless they're in the presence of a matrix. So this is true for all biopolymers, for proteins, nucleic acids, glycoproteins. So the matrix itself, there are a number of matrix compounds. They're all small aromatic weak acids. They've been discovered empirically. There is some understanding of what makes a good matrix, but the mechanism by which the matrix ionizes and then this gas phase charge transfer, it's not really understood. It doesn't really matter because the charge state, there isn't really any information in the charge state of a MALDI ion. So it's not like electrospray where this mechanism is very important. In MALDI, it's not really very important. It just works, and that's all you need to know about it. The key thing about matrices is you need to use a different matrix depending on what it is you're analyzing. And I've shown some of this in the table on this slide. You have to choose the right matrix. If you choose the wrong matrix, you will not see any of your analyte at all. So the choice of matrix and also how you prepare the crystals, this traditionally is something of an art in MALDI, but there are matrices where there's no real skill required, and they work very, very well. But just take this away, that you have to use the correct matrix for whatever it is you're analyzing. And there is another aspect about matrix crystals, and I've shown it on this slide here. This is a very popular matrix. This is alpha cyano. It's very widely used, and you can see it gives you uniform crystals. So wherever the laser spot lands on this sample target doesn't really matter because you're going to get the same signal. However, if you compare this with this matrix, which is synapinic acid, you will see the matrix crystals have this kind of feather-like morphology. And it's necessary, in order to get any signal, it's necessary to steer the laser onto the base of these crystals. And there are several hot spots, several sweet spots in this sample where you may get a signal. But if you're in the wrong position, you won't get a signal. And this is important because one of the advantages of MALDI is you can automate it, but you can only automate it if you have this uniform crystal. So for alpha cyano, automation is straightforward. For synapinic acid, you cannot do this. So analyzing a large number of samples manually turns out to be quite tedious and quite unreliable. But these are some of the very commonly used matrices. Alpha cyano, as I mentioned, this is very popular. It works very well for peptides, also nucleotides. For nucleotides, this one, 3-HPA, is very popular. For proteins, as I mentioned, this one, synapinic acid, is very good. Also, ferulic acid is very good for intact protein analysis. They have different solvents. Alpha cyano, the solvent is acetonitrile. For some of these, the solvents for DHB, the solvent can be water. DHB is if there is such a thing as a generic matrix, DHB is probably it. It can be used for more things than most matrices, but you must choose your matrix carefully. So how do electrospray and MALDI compare? Generally speaking, if you ask somebody who works on electrospray, they will tell you this is the best technique. If you ask someone working on MALDI, they will also tell you that that is the best technique. I think I'm probably unique in that I've spent about 10 years working on MALDI and about 16 years working on electrospray. And I would consider myself to be an expert in both. And I think I'm probably uniquely qualified to actually comment on how these two techniques compare. So Main differences in terms of sample introduction: in electrospray, it's a liquid sample introduction. In MALDI, the sample introduction has to be solid. Electrospray is rather intolerant to salts, but in general that doesn't matter because almost always your electrospray mass spectrometer will be connected up to a chromatography system. So the desalting will happen there. In MALDI, you don't need to do that. It will tolerate relatively small amounts of salt. Electrospray ionization, I would describe this as a completely generic technique. It works extremely well with almost anything you want to analyze. As I've mentioned, in MALDI can work quite well, but it is matrix dependent, so you must choose the correct matrix. In electrospray, the speed of electrospray analysis is dictated by the LC system. So if you run a rapid gradient, you can do your analysis in 1 or 2 minutes. You may choose to do your analysis over a couple of hours. So it's relatively slow. In MALDI, all you're doing is firing a laser at a spot, and you can do that in a few seconds. And so your acquisition is lightning fast. Electrospray, LCMS systems tend to be more complex than MALDI, and it's not because the mass spectrometer is more complicated. It's basically you have a liquid chromatography system coupled to that, so it's generally a more complex experimental system. MALDI, there is very little to go wrong in a MALDI mass spectrometer, so it's a very robust experimental system, and the operation is quite simple, which can be an advantage. In terms of sample purification, electrospray, the sample purification is in line. If you're going to do sample purification, and remember, mass spectrometry works better the purer your sample is, in MALDI you're going to have to do that sample purification offline, and that's quite common. This is important. Electrospray performs extremely well on complex samples. I would say the vast bulk of interest in biological mass spectrometry is analysis of complex samples. And this is where MALDI falls down. MALDI does not perform well on complex samples unless you have pre-fractionated your sample before it goes into the MALDI. The charging mechanism electrospray is different. We see this characteristic multiple charging of species above about 600 Daltons. In MALDI, MALDI ions are invariably singly charged unless they're extremely large. Because of this multiple charging, in electrospray we need to deconvolute the spectra. In MALDI, there's no deconvolution required. Electrospray, the data interpretation, a lot of it's automated now, but it can be quite difficult. In MALDI, data interpretation is relatively easy. The high mass performance of electrospray is fantastic. In comparison, I don't care what anyone tells you, the high mass performance of MALDI is very poor. What you will see is MALDI instruments generally are not used above about 20,000 Daltons. When they are, the mass accuracy is poor, and also the signal intensity is poor. And I certainly would not recommend MALDI for analysis of large biomolecules. I've said here that no imaging is possible by electrospray. For standard electrospray, that's true. There are some recently emerged techniques like desorption electrospray, which can be used for imaging mass spectrometry, but I'm not going to talk about that today. MALDI is really purpose designed for imaging, and we'll be talking about that later on. So what applications are suited to MALDI? So it is ideally suited for high throughput analyses because it's very fast, and typically characterization of small molecules which are already pure. So typically MALDI TOF would be used for quality control of synthetic peptides and synthetic oligonucleotides. It's also ideal for any applications that I would describe as non-research, where the fact that it's very simple and there's very little to go wrong with it is extremely important. So you can transfer this into, say, a clinical setting. I said MALDI is not really suitable for complex mixtures. You can generate very high quality proteomics data from one of these instruments, a MALDI TOF. It's an MSMS instrument. But to do it, you need to interface your MALDI instrument with a nano HPLC. And you do this via this gizmo here. This is a MALDI spotting robot. And I think we have a video here which you can watch. However, the problem with spotting robot, it's just an additional complication. It's an unwanted level of complexity in an experiment. If you're doing a proteomics experiment with a very long gradient and you're looking for reproducibility, that's going to be much harder to achieve with a spotting robot interface with your MSMS. And it's not really necessary because if you compare it with electrospray, that interface is already there. So it's just unnecessarily complicated. And because of that, I would say these systems have pretty much universally been eclipsed by nano LC. However, whereas electrospray, I would describe it as a generic technique, and I wouldn't describe MALDI as a generic technique anymore. I would describe it as a niche technique. However, there are three areas in which MALDI is becoming increasingly important. And as I mentioned, these areas are clinical diagnostics, MALDI tissue imaging, and imaging mass cytometry. So let's talk about MALDI in clinical diagnostics. So as I hope I've indicated, a MALDI TOF instrument, they are a benchtop instrument, they're relatively cheap, they are fast, and they are reliable, and they're very simple to operate. So they are ideally suited for a clinical diagnostics environment. And one company, Bruker, have really been able to exploit this. So they have developed an instrument which they call a biotyper, and it's used for bacteriological identification. And what you will see in mass spectrometry is there is a lot of emphasis on very complicated, very expensive mass spectrometry systems suited for research and suited for quantitative proteomics. But I wouldn't necessarily say that is the future of mass spectrometry, because very few labs actually have the money to operate one of these instruments. What Bruker have done is they've gone in the opposite direction. They've found a mass spectrometer that can be produced cheaply. It can be produced in large numbers, and it's also easy for non-specialists to use it. And I think really this may well be the future of mass spectrometry, where every lab just has a small, simple to operate mass spec in the corner of the lab. Everyone knows how to use it, and it's an essential lab tool. But this is how the biotyper works. So this is really quite nice. It's very simple. I'll just get my pointer back. We start with a bacteriological colony. You can just pick one of these colonies off, and you spot it onto your target plate. And onto your target plate, you add your matrix solution. In this case, it's alpha-cyanosinamic acid, and that's dissolved in acetonitrile. The acetonitrile will have the effect of lysing your bacterial colony and lysing the cells, so you will have a protein solution spotted on each spot of the moldy target plate. You can then process as many as you like. This would be a 384 well plate, so you could put a lot of samples onto this plate. You could easily automate that with a robot. It then goes into the mass spectrometer. The laser fires on each spot, and it will generate a spectrum. And then each of those spectra can then be used in data evaluation. And so this is very easily automated and very high throughput. So how this works is all bacteria are made up of proteins, as is all living things, and the proteins are homologs of each other. So any particular protein in a bacterial species will have a homolog in another species. So it will do the same job, but it will have probably a different sequence. So all of these homologous proteins, they will have slightly different sequence. And because they have a different sequence, they will have a different mass. And this is a natural thing due to evolutionary divergence. So what the TOF spectrum does is it generates a mass fingerprint for each sample, and that fingerprint is unique for any particular species or any particular isolate of bacteria. So I've shown 4 examples here of different bacterial species. This is the fingerprint, and you can see well, some of the masses are similar, like these ones. Some of them are different. Okay, it's not necessary to know what any of these peaks actually represent. It doesn't matter. It's just a fingerprint, and that fingerprint is hopefully unique for any particular isolate or any particular bacterial species. And Bruker actually have a library of bacterial fingerprints, and you simply buy that library and you match your data to the library, and it enables you to identify the species or identify the isolate. It's very quick and it's very, very easy. I now want to talk about MALDI imaging. So in MALDI imaging, this is a technique that's been around for a long time now. I would say probably over 20 years, originally developed by Richard Caprioli and Marcus Stöckli, but it is becoming quite mainstream now. And how it works is your standard MALDI laser is a 337 nanometer ultraviolet laser, and it will irradiate an area on the target round about 50 nanometers in diameter. And your MALDI sample is positioned into the path of the laser. Remember, we steer the sample onto the laser rather than the laser onto the sample. But this is done just using an XY stepper motor and a robotic platform. And that's done with the aid of a source viewing camera. And what that means is every spectrum that you generate in a MALDI analysis, it has not just the mass data and not just the spectrum, but it also has the positional information, the 2-dimensional positional information for where that spectrum was acquired from, where the laser sample hit the target. So you have this 2-dimensional positional information. So what you do is you can prepare a tissue section, and this tissue section is then prepared onto the MALDI target plate. You can spray it with a matrix solution, and then you use your laser, and you can use a very high repetition rate laser. Instruments are specifically designed for this. Will have a 200 hertz laser, so it's firing laser extremely fast, and it can perform a raster scan across the target plate, and this will generate an image of the tissue, and I will try and show you that. So what you get is you get an ion intensity picture for each MZ value, and this will show you the distribution of discrete compounds within your tissue section. Of course, you can also do sequential tissue sections, and this will allow you to build up a three-dimensional image, as you would do in classical histology, and you can look at the three-dimensional distribution of not just one compound, but multiple compounds in your sample. So this is an illustration of what I mean. So this is 3D MALDI imaging through a kidney. So this is a section through the kidney, and you can see the kidney is divided into different regions. So this is the cortex of the kidney, this is the medulla of the kidney, and if we choose a particular MZ value, and here on this slide the MZ value is 7,901, you can see how this is distributed within the tissue, and you can see that this particular MZ is occurring only in the cortex, and it's more or less absent from the medulla. So in this particular experiment, they did 120 serial sections. The mass range, pay attention to that, because the mass range is, well, this is a mass range of large peptides and small proteins. So this goes up to 20,000, but significantly, it doesn't really go above 20,000. And the resolution of the image is 50 microns. And I've got another YouTube video which I'd like to show you.
The following video shows a screen recording from the Flex Imaging software. It shows a MALDI imaging dataset of a mouse pancreatic tumor acquired on a Celerix FTMS. After the MALDI imaging measurement, the same section was washed, then stained by H&E. A virtual microscopic slide of this stained section is then co-registered with the MALDI imaging data. A portion of the tissue was imaged at a spatial resolution of 50 micron. We can see from the gross anatomy that this imaged area encompasses several anatomical features, including a lymph node and portions of the tumor. A variety of mass filters can be selected, and the presence of that mass shows different distributions across the tissue. Selections of different masses show different distributions that can then be correlated to the anatomy. One mass marker in particular shows an unusual distribution across the tissue. If we look closely, we can see the presence of some darker areas that appear to be green dots. And the question can be asked, from what structure do these green dots arise? Once the image is co-registered, the Flex Imaging software allows for seamless fading between the mass spec signals and the virtual microscopy slide. If we overlay the virtual slide with the mass signals and zoom in, we begin to see additional detail in the tissue. From here, it becomes apparent that the green mass signal is always found in epithelial ducts. We need microscopic resolution in order to identify the specific arrangement of cells that form the epithelial ducts. Zooming in to a region containing ducts shows that the mass corresponding to the green signal is present in all of them. Additional correlation can be made through seamless fading between the mass signals and the microscopic image. At first sight, the global average spectrum does not appear to show many high-intensity signals. However, as we zoom in on the baseline, we begin to see the presence of many low-intensity signals. This is due to both the high mass resolving power and the high dynamic range of the FTMS instrument. Zooming in on a single nominal mass shows that it actually contains many different mass signals, which are very closely spaced. Frequently, these mass signals show different distributions across the tissue. In this example, we see 3 peaks separated in mass by less than 100 millidaltons, which show completely different distributions across the tissue, giving us ultimate specificity.
So that's an example of MALDI imaging. I think it's a very interesting technique. It's certainly becoming more popular. However, MALDI imaging suffers from all the same limitations as all MALDI, and those 2 limitations are basically poor performance for complex samples and also poor performance at high mass. And there is a more recent technique called imaging mass cytometry, which overcomes some of these. So I guess most people are familiar with flow cytometry, and I find this is useful to understand this technique before looking at imaging cytometry, mass cytometry. So basically in flow cytometry, you have a cell suspension. You incubate your cell suspension with a specific fluorescently labeled antibodies. Your cells are then going to pass across a laser in single file. The laser is going to illuminate those fluorescent antibodies, and there's some kind of sorting mechanism which allows you to identify different populations of cells based on the labeling of specific antibodies. So in mass cytometry, the principle of this is really very, very similar. What you do is, instead of using fluorescently labeled antibodies, you use mass tagged antibodies. So you can have up to 100 different antibodies which are tagged with a rare earth metal. So these are lanthanides. There are roughly 100 lanthanides in the periodic table, and they can all be used as individual mass tags. So the signal from each different lanthanide will indicate the presence of a specific antibody. So you incubate your labeled antibodies with your cells. Your cells are then sprayed as single cell droplets, and then they pass into this 5,000 degree plasma, and that will vaporize the cells, and all that there will be left will be these lanthanide ions, and they can be quantified simultaneously in the mass spectrometer. So this is your workflow. Here's your cell. You label it with your mass tagged antibodies. They go into the nebulizer, one cell at a time. They're then vaporized, then pass through the quadrupole. You have a time of flight analyzer, and that will measure simultaneously measure the different lanthanides and quantify them, as you can show here. And this will generate something rather similar to a FACS plot for your cell type. If anybody's interested in this technique, we do have this, not in our lab, but they do have this in the Kennedy Institute. So that is mass cytometry. A development from that is imaging mass cytometry. And so what you do here is you work with a paraffin embedded tissue. You incubate it with your mass tagged antibodies, these lanthanide labeled antibodies, and instead of them passing directly into the mass spectrometer, what happens is you have a laser ablation cell. So your tissue section passes through this cell. The cell has, so the laser hits it, but it passes through a window, which is a single micron across, and this will raster through your tissue sample and generate an image. So this is the workflow. This is your tissue. You incubate it with up to 100 different antibodies specific for different proteins. Your laser rasters through the sample, ablates it, and then generates an image, and you can do multiple sections to get a three-dimensional a profile of your tissue. So again, this is the workflow. We have up to a 200 Hertz laser. It's taking a sample every 1 micron in your tissue. It's then generating, quantifying the different lanthanides present in that sample, and then mapping it onto the tissue. So these are some examples of imaging mass cytometry. I really think these are quite beautiful. So if we look on the left here, this is the total ion current. So this is all the signal that is coming from the mass spectrometer plotted onto the XY axes to give you this image. And so you can clearly see there that here's the tissue, and this looks like an epithelium, and you can see different cell types and different morphologies within this image. And then overlaid onto that, we have antibodies for 3 different types of molecules. So we have antibody for histone H3, that's in red, antibody for DNA in blue, and beta-actin, so that's a structural protein, sorry, that DNA in green, and beta-actin in blue. And you can see this is your, I guess this is your basal lamellae of the tissue. The same tissue, you can, because you can do this with a large number of antibodies. In the same tissue, we have different patterns. So we have 3 different proteins here, E-cathedrin, collagen, and alpha SMA plotted on this tissue. Again, this is a different image. This is a section. This is the total ion current. Again, we've got beta-catenin in red, histone H3 in blue, and alpha SMA. And then here, a different selection of 3 different proteins, pankeratin, vimentin, and DNA. So there are lots of advantages to this particular technique. So that's really all I want to say about MALDI. Just to summarize, think of MALDI it's an alternative soft ionization technique to electrospray, and it's different in that it generates singly charged ions. The matrix needs to be specific for whatever it is you're analyzing. So it's not a generic technique. You need to choose the right matrix. There isn't very much sample preparation necessary. The analysis is extremely fast. As I've said a number of times, that MALDI, it's not suited for complex analyses, and it's also not suitable for analysis of large molecules, but it is ideal for small molecule quality control. It's ideal for clinical microbiology, as I explained in the biotyper application, and also in tissue imaging. So we've looked at mass cytometry, and this is a development of flow cytometry, and it uses mass spectrometry to quantify rare earth metals that are coupled to antibodies which are bound to cells. You can quantify a large number of species simultaneously, which is very advantageous. The development of imaging mass cytometry, this gives you spatial information to multiplex mass cytometry. So by multiplex, I mean this ability to analyze 100 different protein types in a single experiment. And the resolution, I think, is superb. The resolution is 1 micron, which is comparable to optical microscopy. So one advantage of imaging mass cytometry is you can perform it on paraffin embedded tissue sections. These can be historical samples. So you could do this on a sample from a patient who died of a cancer many years ago. But you can go back to that sample, you can interrogate it by imaging mass cytometry, and you can build up an experimental data set on historical samples. And you will know, because it's a historical sample, you know what the clinical outcome of that particular patient was. And I think this is extremely powerful. And I really do believe that imaging mass cytometry will completely transform histology. That's all I want to say about MALDI. It's all I want to say now about our training seminar. That's the last one in the series. I will, if anyone's in, all of these lectures are, they will be available online, so you can watch them again if you want to. I will produce a quiz which you can have a look at just for a bit of fun, and you can see which ones you might need to watch again. And that's it. I'll take any questions now if there are any. And I'll stop sharing my screen.
I don't have a question, but I'd just like to thank you for running the series.
I hope you found it useful, Lucy.
Yes, it's been very useful, thanks. Yeah,
Excellent.
Particularly today's, thanks.
Good, I hope you get a chance to use it.
Yeah, me too.
Okay.
And thanks too.
You're very welcome. You're welcome. I've got thumbs up. Thank you very much. Okay, I'm going to end the session, and thank you very much for listening.